Genetic engineering will benefit future generations through the advanced treatment of disabilities, and the article Germline Gene Therapy Safety from Science Daily, published on the 3rd of June 2019, issues this problem with its benefits and consequences, along with some major breakthroughs in gene therapy, such as the world's first gene-edited babies in China. Dr. Mitalapov, the director of the OHSU Center for Embryonic Cell and Gene Therapy, mentions that germline gene therapy holds enormous promise in preventing inherited diseases, yet this practice also contains many ethical issues as this therapy is generational. The more than 30 countries, including China, already having regulation and laws in place prohibiting genetic modifications to the human germline. This makes germline gene therapy a highly ethical medical technique. So what is genetic therapy? This video highlights the definition of gene therapy and its different methods.
What is cell and gene therapy? Cell and gene therapy uses minute pieces of our cells and genes, and sometimes both, to repair problems that may cause disease. It is giving hope for patients for whom treatment has failed or doesn't exist yet. For example, children born with a fatal or life-threatening condition resulting from a single faulty gene. The problem can be fixed by delivering a new functioning version of the gene into the cell. In some cases, cells from the patient are taken from the body, engineered with a new gene, and put back again. In other cases, it's possible to deliver the new gene directly into the place where it's needed in the body. In some illnesses, such as cancer, the immune cells can be upgraded to better recognize the rogue cells and kill them. These are known as CAR T cells. The exciting thing about cell and gene therapy is that it fixes the problem at source, hopefully for good. The prospect of a permanent cure rather than a lifetime of treatment is a paradigm shift for modern medicine.
There is still much to learn about the subject, but many advancements in the prevention of genetic diseases and viruses have already been made. However, gene therapy can also raise problems by causing unwanted immune system reactions or targeting the wrong cells, which causes tumors. With gene therapy, there is also a chance that the virus could adapt and continue to cause diseases. Due to this treatment option being risky, it is still under study to make sure that the therapy is safe and effective. Gene therapy is currently being tested only for diseases that have no other cures. The main topic of this video is germline therapy, also known as germline gene therapy, which is a type of therapy that focuses on reproductive cells which are known as gametes. To briefly summarize gametes, they are sex or reproductive cells that contain half the required genetic material needed to form an organism. They are formed through the type of cell division called meiosis. These cells can either be male or female, and one from each gender are needed to fertilize and to form a new cell, which is a zygote, which contains two halves of genetic material, one half being from the male and the other being from the female. Germline therapy is carried out with the intention of changing DNA and passing it on to the next generation. This type of therapy allows the correction of disease-causing gene variants and mutations. Through germline therapy, future generations will have modified genes which will prevent them from being affected or contracting a genetic disease.
Genetic therapy will be even more beneficial for the future generations as the treatment for genetic disabilities and diseases become more advanced. Gene therapy clinical research was first approved to be conducted by William French Anderson, who is considered to be the father of gene therapy, and regarded as one of the major pioneers of genetic engineering. The patient receiving the treatment for a genetic defect was a 4-year-old named Ashanti De Silva. This genetic defect left her with ADASID, a severe immune system deficiency. Even though Ashanti's immune system was partially restored by replacing the defective gene with a functional variant, the effects of the therapy remained temporary.
We know that the idea behind gene therapy is to replace faulty genes with functioning and healthier versions of them. However, DNA can't be injected into a cell the way vaccines are injected into humans. Instead, this new DNA needs to be integrated into a cell using a vector which is a carrier for the DNA. The vector acts as a vehicle to carry the new DNA into the cells of a patient without it being broken down. Once inside the cells of the patient, the DNA produces therapeutic protein and treatment for the patient's disease. Vectors are chosen based off the type of cell that will be modified and the size of the cell. Plasmids are the most common type of vector. They are normally used for the modification of a code of bacterium. Plasmids are used for bacteria because bacteria also have plasmids, as well as mechanisms that can help share them. Other examples of vectors are viruses, stem cells, and liposomes.
The second vector is viruses, and they introduce DNA by invading cells. So using this concept, viruses are strategically engineered to not cause any harmful effects, and to only introduce the foreign DNA to the target cells. It is crucial that the new gene reaches the correct cell because delivering a gene to a wrong cell will be ineffective and could trigger a tumor. The third vector is stem cells, which are cells which all other cells in the human body are created. In laboratories, stem cells can be trained to help fight diseases. The fourth vector is liposomes, which are fatty particles that can carry new therapeutic genes to the target cells and pass the genes into the reproductive cell's DNA. Gene therapy in general has led to successful results against certain diseases such as leukemia, hemophilia, and severe combined immune deficiency. An example of a disease that could be cured is cystic fibrosis, which is the most common genetic disease in Australia. Cystic fibrosis has many effects on bodies, such as clogged airways caused by thick mucus in the lungs, making gas exchange difficult. A person with cystic fibrosis is homozygous for a recessive allele called CFTR, and since 1989, scientists have investigated potential ways of transferring normal CFTR genes into human lungs. However, several significant barriers prevent gene therapy from progressing and becoming a reliable form of treatment. If we look more closely at germline therapy, it is still not ready to be used on the public, given the uncertainty of the treatment and the chance of it having negative effects on patients. There is still a lot of progress that needs to be made in this particular aspect of gene therapy, which is why it is not ready to be made available to the public.
Germline therapy affects both genders because the therapy acts on gametes which are present in both men and women. Adults would be the ones who are affected by germline therapy since they would be the ones who are looking to have children. This group of adults can be further pinpointed to those who have genetic disorders and diseases which have been running through their family for generations. Through germline gene therapy, they would look to remove the problem from their gene flow through the insertion of new genes, allowing them to have offspring who will not have the same problem as they or their older generations did. One of the issues that we have with germline therapy and gene therapy is that as much as it is used for curing people of their diseases and disorders, there is also a chance that the treatment could have a negative effect on them. In germline gene therapy, there are many challenges with delivering a gene into the sperm or ovary cells. These include inserting the gene into a chromosome without disrupting other genes, delivering genes without causing immune reactions, as well as being able to turn it on to produce the correct protein. Something that also must be taken into account is the fact that a disease has the potential to build a resistance. If the subject is looked at from an economical perspective, it can also be seen that genetic treatment would prove to be costly both for the further development of it and after it has been developed and made available to the public.
Genetic treatment in general could turn out to be a massive legal issue. Considering the fact that the treatment is so expensive, it is hard to believe that future parents wouldn't be upset if they paid lots of money for it to turn out wrong, and maybe even for one of them to possibly come out of the treatment worse than they were before it. To illustrate, it would be hard to imagine a person not getting angry if they or their spouse received a tumor from an incorrect injection of a gene into their body. The mistakes made in gene therapy could prove to be too dangerous and could potentially lead to many legal problems to institutions and gene clinics that choose to engage in this method of therapy.
Some further questions that we have on the topic include: when will germline therapy become available to the public? What do scientists believe is the correct approach for ensuring that the genetic modifications made during the treatment are long-term? And lastly, if germline therapy does become a successful form of therapy, then what sort of rules would we need to enforce to ensure that the chance of a person being negatively affected from the treatment is limited?
Overall, germline therapy is an upcoming form of genetic engineering that will pave the way to more future forms of medicine. Even though it is ethically unaccepted by people nowadays, germline gene therapy will produce many outstanding benefits that can benefit future generations in the future.